L9c continues with DNA metabolism, and for our third part, we're talking about correcting mistakes and mutations. So part A will be 3' to 5' exonuclease activity during replication. B, base excision repair, DNA glycosylases. C, nucleotide excision repair. D, double strand breaks. And E, mutagenesis and the Ames test. So DNA editing and mutation. So during replication, DNA polymerase, with adding of nucleotides, there is a mistake about 1 in every 1,000 nucleotides. So about 1 in 1,000 nucleotides are incorrectly added due to alternative base pairing due to keto-eno-tautomerism. The DNA polymerases delta and epsilon have 3' to 5' exonuclease activity, and they decrease the error rate from 10 to the minus 3, which we have over here, 1 in 1,000, to 10 to the minus 6 would be 1 in a million. So that's a huge decrease in error rate because of our 3' to 5' exonuclease activity. And then we have another kind of editing ability, which is called post-replication mismatch repair, and it increases our DNA fidelity from 10 to the minus 6 up to 10 to the minus 10th. So the final nucleotide error rate within our DNA sequences are about 10 to the minus 9th to 10 to the minus 10th, and that's accurate. Very, very precise. So here's an example of error correction by 3' to 5' exonuclease activity of DNA polymerase 1. So this is the prokaryote, but it's also true for the eukaryote. So here's the DNA polymerase, DNA active site, and here's our 3' to 5' proofreading exonuclease active site. So C is a rare tautomeric form of cytosine that pairs with A rather than a G. So usually we have CG base pairs. Now we're going to have a CA base pair, which one of our ketones becomes an enol that pairs with A as incorporated in the growing strand. Before the polymerase moves on, the cytosine undergoes a tautomeric shift back to the regular C, and the new nucleotide is now base paired with the A, and this doesn't fit into the active site correctly. The mispaired OH of the growing strand blocks further elongation. DNA polymerase slides back to the position of the mismatched base pair, so it slides back one position, and now this mismatch is in the editing site rather than the polymerization site. The mispaired nucleotide is removed. The DNA polymerase slides forward and resumes its polymerization activity, and now we add the correct base. So the correct base is now between an A and a T rather than an A and a C. So that's 3' to 5' exonuclease. So upon polymerization, polymerase pauses because these tautomers don't last very long. They're ephemeral type structures. The normal structure is much more stable than the abnormal tautomeric structure. And what was a normal base pair, because we shift back to the normal tautomeric structure, is now a mismatch, and our 3' to 5' will remove that mismatch, and we will try again to add the correct nucleotide. So now let's talk about mismatches. So when we have a mismatch, the DNA bulges, and we need a way of figuring out which one is wrong. With the bulge, which one is wrong? The parent DNA often has methyl groups on it. The newly synthesized strand tends to be unmethylated, and that way the repair mechanisms know which one is the old strand, which one is the new strand, and the assumption is the old strand is correct and the new strand is wrong, and you remove the mismatch from the new strand, and then you correct it. And we're going to talk about how we do this correction. Here's our normal DNA. Everything's okay. Sequences are okay. Base pairing's okay. Then we have a damage to one of the bases, and we have two strategies for removing the incorrect base. We can have what's called a glycosylase, and the glycosylase directly removes the incorrect base. That leaves the phosphodiester strands okay, but we're missing a base. We have what's called an incision endonucleotide, and it will cut right here. And then we have an excision endonuclease, and it will remove a couple of these bases. Then a DNA polymerase will come in and extend from here. It extends over. And then DNA ligase will ligate the chains, the two pieces back together, and we will have corrected our mistake. Or we can have another DNA damage, and we can have the incision nuclease, and it will directly cut rather than remove the base. You can directly cut, and then excision endonuclease creates a gap so the DNA polymerase can extend. So this is our 3' end. We can extend over to here, and then we DNA ligate the two fragments together. What determines which one of these is used depends on what the damage to the base is. There's several strategies for different types of mutations or different types of errors that determine whether you use a glycosylase or whether you use the incision endonuclease. You may have wondered why we use thymidylate rather than uridylate. So we use T rather than U, and the reason is the C has an amino group that over time will spontaneously deaminate to form uracil. So C is slightly unstable with time, and it will release an ammonia group, and then it becomes a U. So any U that's found in DNA came from C, and we know that that needs to be fixed. That needs to be replaced. So rather than U, we use T. So when we get this spontaneous deamination from C to U, we know that U is a mutation and should be corrected. So we spontaneously convert C into U. And it's a small amount of C that's undergoing this, but it's significant as far as your DNA code is concerned. And then we have a uracil DNA glycosylase that removes a U. Again, we have an endonuclease that gives us two DNA fragments. We have a DNA phosphodiesterase that generates a gap. And now we fill in the gap with DNA polymerase B ligase. So here we've added the C and we've ligated the two pieces together. So the key point is C spontaneously mutates to a U. We take out the U. We cleave the phosphodiester backbone. We generate a gap. We fill in the gap. And then ligase puts our two pieces together. And now we have our correct G-C base pairing. So excision of pyrimidine dimers. And what we're showing is two thymidines. You can tell they're thymidines. Here's a CH3, there's a CH3. And notice in the DNA double helical molecule, proximal thymidines, they almost stack directly on top of each other. They're only slightly offset. And with ultraviolet light, we form what are called cyclobutane rings. So this double bond and this double bond, the orbitals between this double bond and this double bond will overlap, and the light causes formation of covalent bonds, and this is called a thymidine dimer. It'll also happen with TC and CC, but the overlap of the molecules and all is more optimal with the thymidine, and this is a very serious mutation. And we have an enzymatic activity that actually removes the TTs and generates a gap. DNA polymerase and ligase fill in the gap, and now we have AATT, except the TT is not covalently linked. So we've corrected the thymidine dimer into thymidine, correctly base pairing with A and T. There's a genetic disease called xeroderma pigmentosum, and xeroderma pigmentosum is where we have defective editing to remove thymidine dimers, and these people tend to have high rates of skin cancer because where are you getting exposure to ultraviolet light? Well, it's in your skin. So a defect in the ability to edit out the thymidine dimers will result in skin cancer. Chromosomal damage, repair of double-stranded DNA breaks. Occasionally they break from High energy radiation, sometimes just mechanical shear during meiosis and mitosis can break chromosomes. Frequently though, it's high energy radiation. If this is overlapped, these are called sticky ends. If they're overlapped like this, we have exonucleases that whenever they see these overlapping structures like that, they cleave them off until the DNA strands are equal. So loss of nucleotides due to degradation from the ends. So our free ends like this are really unstable, and we'll degrade those ends until they're flush with each other. And then we have what's called non-homologous end joining. We will actually reconnect the broken chromosome. Although when we've done that, we've lost genetic material when we reconnect our chromosomal fragments. However, if this is happening during a time in the cell where we have a nearby homologous chromosome or a sister chromatid, for instance, during meiosis or mitosis, we can use the sequence from the other chromosome to fill in our gaps. So that's called a copying process involved with homologous recombination. So if we have a homologous chromosome around, or even a sister chromatid around, where we have DNA sequences on the other chromosome that we can use, we can base pair this fragment, this fraction, we will have the gap, we will have this gap, and we can fill that in. So we'll end up with a complete sequence restored by copying from a second chromosome. So this is called homologous end joining. So if you had a choice, you would want to do homologous end joining because there's no loss of DNA sequence. Over here, we've lost DNA sequence. So now that we've talked about mutagens and mutagenesis, etc., mutagenesis and mutations of our DNA is one of the major causes of cancer. So about 35 years ago or so, a guy named Bruce Ames developed an assay to determine are certain chemicals mutagenic. Because that's good to know. It's good for industry to know. It's good for the consumer to know that certain things that may be in your diet or certain things in your environment have a tendency to cause cancer, and those particular substances are mutagenic. They cause mutations in our DNA sequence. So this is the Ames test for mutagenic substances. And what we do is we've engineered a salmonella bacteria that's unable to synthesize histidine. And for this bacteria to grow, normally for it to grow, it has to have histidine in its nutrients. You inoculate plates with tester strain histidine oxytrope, which means it has to have histidine in this media to grow. So if we inoculate these bacteria on a plate that doesn't contain histidine or contain low levels of histidine, we'll get a little growth and then it will stop. But if we've added a filter disc containing a test chemical, and the test chemical happens to be mutagenic, then we will get many, many colonies of the bacteria rather than just a few. And these colonies are called revertants. They've reverted back to a wild type that can make histidine. So in order for them to grow, they've had to make histidine. So this is an indication that the test chemical that was put on this piece of filter paper was mutagenic. And it mutated the defective histidine gene back to a histidine gene that can synthesize the histidine. So if this contains the test chemical, what we see is the test chemical will diffuse into the plate. And so this is where the test chemical concentrations were really high, and it is actually killing the cells, but eventually it radiates out by diffusion, and we start to get revertant cells. So this is actually a high number of cells that can now make histidine. So negative result, positive result. If the chemical is non-mutagenic, it will look like the negative result. Here's our normal plate. Here we put in the filter that contained the mutagen. This is the highest concentration of mutagen, and notice it has to diffuse way out quite a ways before we start seeing the revertants, and that's because in here it's actually toxic. It's only here that we get revertant colonies, so this is actually killing the bacteria in this stage. And here's our next highest concentration. So this is the highest, this is the next highest. It doesn't have to diffuse out as far before we see revertance. And this is the lowest concentration, and we immediately see revertance because it's a mutagen rather than being a bactericidal. This is like bactericidal. This is killing the bacteria. This doesn't kill it quite as much. This kills it less. So this is the end of L9c.